Hello, my name is Danielle Hohman, and I will be presenting my research on the role of DDX5 in HPV-positive cancers. I plan on giving an overview of human papillomaviruses, head and neck cancers, the family of dead box helicases, along with DDX5, which is our protein of interest. After that, I'll be discussing some of the results that we have seen thus far and our plans for future experiments. Human papillomaviruses are small, non-enveloped viruses with a circular double-stranded DNA genome. The early genes encode for proteins that are important for viral expression, replication, and survival. In high-risk HPV, the second and third oncogenes, E6 and E7, are the major drivers of cancer. They block, degrade, and inhibit tumor suppressor genes. Transmission can occur by either sexual contact or non-sexual routes, like through vertical transmission from mom to newborn, or by fomites like undergarments. HPV is the second most frequent cancer in women. It causes cervical cancers and accounts for 200,000 cases of precancers yearly. Among the 30 known HPV types that infect the cervix, vagina, vulva, penis, and anus, about 84% of the invasive cervical cancers are caused by the high-risk subtypes HPV-16 or HPV-18. For this research, HPV-16 was used. Head and neck cancers are the fifth most frequent cancer worldwide, 6 in the United States. The risk for them increases for individuals over the age of 50 and has a 50% survival rate. Majority of these cancers are known as squamous cell carcinomas, and they arise in the upper level of the aerodigestive tract. More specifically, the mucus-lined cells in the mouth, throat, and nose. Typically, head and neck cancers are seen in response to smoking and alcohol use, but HPV-16 infection is resulting in increased cases. This being said, there are two types of head and neck cancers, HPV positive and HPV negative. HPV positive is a result of HPV infection and independent of tobacco or alcohol use. These are seen through associations with oropharyngeal squamous cell carcinomas. On the other hand, HPV negative is a result of a loss of function mutation in p53 or other tumor suppressors due primarily to tobacco or alcohol use. The dead box helicase family is a large family that aids in multiple aspects of RNA metabolism, ranging from transcription to degradation. The proteins in this family are highly conserved. In their helix core, they share 9 conserved domains. The helix core contains an ATP binding and hydrolysis domains, along with RNA binding and helicase domains. As a result, they're able to bind RNA and unwind it. The spaces found between these motifs, along with both the 5' and 3' ends, give each individual member their substrate specificity. This family of helicases acts differently than conventional helicases. Panel A of the figure below shows a conventional helicase that can bind long sequences and unwind. Panel B, on the other hand, shows the mode of unwinding for a dead box family helicase. They unwind short local sequences in a non-specific manner. Dead box helicase 5 is a member of this family. It's mostly localized to the nucleus, but it can shuttle to and from the cytoplasm. To do so, it contains 2 nuclear export signals and 2 nuclear localization signals. When in the cytoplasm, it acts as an ATPase. On the other hand, when in the nucleus, it plays a role in transcriptional coactivation, RNA unwinding, and alternative splicing. DDX5 also contains an array of phosphorylation sites, but for our research, we're very interested in the site located at tyrosine 593. Phosphorylation at the site results in cytoplasmic localization. For our experiments, we used both HPV positive and HPV negative head and neck squamous cell carcinoma cell lines. The HPV negative are seen to the left in all figures and are labeled as SCC1, 9, 19, whereas HPV positive are seen on the right and are labeled as SCC 47, 2, 90, 152, and 147T. Our control for these experiments, NTERT, is an immortalized non-cancer cell. To determine the expression levels of DDX5 in the head and neck cancer cell lines, RT-qPCR was performed in both HPV positive and negative cells. This is seen in the graph on the left. A significant upregulation in all cells except for SCC90 was seen compared to the untransformed control. Higher levels in HPV positive were seen compared to HPV negative. To determine if this trend was also noted at the protein level, Western blots were run to view DDX5 protein levels in the same cell lines. Beta-actin acted as our control, and we normalized to DDX5 expression to it. There is no significant difference noted between the HPV positive and negative cell lines. This may indicate that the role of DDX5 might be different in HPV positive versus negative cancers. Since it's known that DDX5 has different roles when in the cytoplasm compared to the nucleus, we were interested in determining the localization pattern of DDX5 in our HPV cell lines. Cell fractionation was done to isolate total nuclear versus cytoplasmic DDX5 protein, and Western blotting was performed to visualize the DDX5. GAPTH acted as our cytoplasmic control, where lamin AC acted as our nuclear. While there is increased nuclear localization of DDX5 seen in both HPV positive and negative cells, the ratio of nuclear to cytoplasmic is significantly greater in HPV positive cells. This brought up the questions of if and how HPV is promoting DDX5 nuclear localization. Since DDX5 is predominantly a nuclear protein that can have cytoplasmic activity, it must be able to exit the nucleus. In order for it to become cytoplasmic, it must become phosphorylated. c-Abl1 kinase will enter into the nucleus in response to growth factors, and it phosphorylates DDX5 at tyrosine 593. This is what induces the export via the RAN-GTPase pathway. In order to test this, immunofluorescence was used to visualize what is occurring in both the positive and negative HPV cells. When the cells were probed for DDX5, we saw the lowest expression of DDX5 in our control, but no cytoplasmic DDX5 was seen in any of the three. There seems to be increased nuclear DDX5 in the HPV positive cell lines than the negative, which confirms what we saw with our fractionation Western. When probed with phosphorylated DDX5, we saw mostly cytoplasmic DDX5 in the HPV negative cells and mostly nuclear in the HPV positive cells. This gives us the idea that HPV is interfering with the export of DDX5 because we see that it is still being phosphorylated but sequestered to the nucleus. Our future investigations include first identifying how HPV promotes the nuclear localization of DDX5. Second, we want to determine the role of nuclear DDX5 in HPV-associated cancers through microRNA regulation and identification of binding partners. Finally, we want to determine the downstream effects of increased nuclear localization of DDX5 in tumorigenesis and the progression of cancer. Thank you.